לא, אני יודע, אבל כאילו, אז ניסיתי לפני שנים, כאילו, פתרון להכיר את זה הקלה.
אני האמת נראה לי כאילו זה מעט אחד, כאילו, כי עברנו לזה, ופתאום כזה, כאילו גם בנחלות הישר,
Okay, שומעים? Okay, שומעים? You're okay? So, I hope you enjoyed the talks by Chaya. I want to start by saying that every year I hope after her talks that some of the computational guys in the audience will pick up the opportunity and go to talk to her or other people in this field, because I don't know what you felt, but I find it kind of almost it's the way this system is being organized is a magic thing. I mean, there's no engineering company in the world that can do 1% of that thing. That each individual neuron will find its way to all these places that it needs to go to and communicate and establish the synapses, and in the right time and in the right place and to the right neurons. And somehow there is, I think, a big barrier between what they tell us, I mean, what we know in general, with all these gradients. It seems to be very, very sloppy. It's like you have some gradient and then the neurons go around the gradient and then they find their way. It sounds kind of very naive in one way, and it seems that I think that there's a lot of theoretical work to be done there on the boundaries of what can be achieved and what's not, and it's in a way, I think, a non-existent field. So I don't know anyone who works on that. I've never seen any model on that, and I think it's extremely interesting, and there's a lot of data to work with and very good models. I mean, experimental models to test it on. So I encourage you to think about it and maybe to go and talk to her, and I think this could be an extremely interesting project. Okay, so today this is something that I flip back and forth every year, more or less, as an introduction to this part of this course. Sometimes I put it at the end of this section and part at the beginning. In a way, this course is called Physiology of the Nervous System, but as we talked about at the beginning, what we're interested in is not strictly just the biology, but also the function. And the function is it's loosely defined thing when we talking about the nervous system. So we're always kind of going on the border between real biology and things that are more computational. So in a way, the other way that this subject is referred to is systems neuroscience. So how many of you took Physiology 1, like the first one? So you had the impression that we're talking about systems. We talk about visual systems, the motor system. Maybe we'll talk later on the somatosensory system, the auditory system. So we're talking about systems, and this is systems neuroscience in the sense that there's some modality or some function, and it's not done by one neuron, it's done by a combination of brain areas and neural networks within them, and we're trying to understand how this system works in a way. So what I'm going to talk to you about now is in a way a bridge between, so it's something that is coherent for all these systems on one hand, and on the other hand is trying to focus us later to the subject that we're going to talk about, which is physiology of single neurons. And it's kind of strange. The connection is not immediate, and this connection is what I'm trying to establish now. Because at least my feeling, and that's the way I think about it, and that's the way I think it seems to happen in this field of neuroscience recently, that the focus is going there in some ways, and I'll try to explain to you what I mean and why it is important. Okay, so I'll skip that. And so imagine that you think about the field of neuroscience. So if you'll ever go to this every year, around 2 weeks from now, there's a big meeting in the United States called the Society for Neuroscience meeting, or people call it SFN. And at some point, if you haven't, has anyone been there? No one. Okay, so what? You're too young. It's okay. By the time you'll finish your PhD, probably at least half of you will be there. And you'll find it's quite amazing. There's about 34,000 people or 35,000 people participating in this conference. And you're coming from this place that is supposed to be at the edge of neuroscience. We're doing the most amazing things. But you'll find out that in this huge space of neuroscience we occupy, I mean, the fields that we are interested in are actually very, very limited. Most of neuroscience is actually talking about this field, which is how to go from genes to functions, or what is the function of specific genes in the brain. And this is a lot of molecular biology and development, and creating mutations and see how they affect the function, or finding a problem in patients and then trying to figure out what is the genetic source of that. So this is hardcore biology, classical biology in many ways, and it is related to neuroscience because the subjects that are working on the groups of genes that are talking about are things that are related to the brain. The other big part of neuroscience is cognitive neuroscience, and this has to do with this bridge with psychology. A lot of fMRI work, EEG, classical cognitive psychology, and somewhere in between there is this field which is called systems neuroscience, which is somehow between these two areas. So it's more animal models, but working on the subject that we just discussed a few minutes ago. And these are the kind of experiments and papers that you are exposed to a lot here. And inside these systems neuroscience is like a tiny field which is called neural computation and neural coding. Okay, it's really, this is the over exaggeration of the thing. Okay, this is so. This is like trying to convey the picture, but the real thing is that you're going into a building which is about the biggest department store that you can imagine. Imagine like you have the whole floor of Azrieli 3 times, all of it filled with posters, and 2 rows of these posters are dedicated to that. Okay, this is about the scale of the thing. Okay. On the other hand, it's really at the heart of everything else, and this is what I'm trying to convey to you now. And the reason is that it doesn't matter where you go from, if you go from the genes or you go from the cognitive or you focus on the systems. You remember that what we talked about is we want to understand how the brain works. We talked about this we talked about this clock idea of how things are working. What does it mean to understand how things work? And eventually, when you go to the brain, you have to talk about neurons and networks, and they are process information in some ways, and this information is, and I'll try to explain to you the whole concept in a few seconds. They communicate with each other, and this is the kind of the thing that we're trying to understand when we are trying to understand how the brain works. Okay, yes?
Can you please turn off the light over there?
This one? Okay? Okay. Right. So, how many of you have computer science background? And the rest are completely, would say, completely ignorant or have nothing? I'll show you and then you'll tell me. So how many of you knows what TCP/IP is? TCP/IP, UDP. Okay, so few knows. Okay, so let's imagine that you have a computer server and you work on your laptop or on your desktop and you want to go to Google and you want to find some information. Okay, so you just type something and you get exactly what you want, but something happens behind the scenes, right? I mean, somehow the things that you type on your keyboard are translated into a way that goes in wires somewhere in the world, bring the information back and convert it into some text or image or something that you can work with, right? So what is working behind the scenes? So you know there are these cables that are connecting your computer to the network, all right? And basically, if you would put an electrode in one of these cables, what you will see is sequence of like, the block, the volt, you'll see voltage, you'll measure voltage, and you'll see the voltage goes up and down between 0 and 5 volts, or 0 and 3.3 volts. This is what you'll see, and it will do it very, very rapidly. You'll have to have a very fast machine if you want to see it. And you see that two things happen there. One, that you have a clock, and the clock is like just ticking just to the clock. So it goes 0, 5, 0, 5, 0, 5 at a regular rate, exactly, very, very precise rate. And then you have another signal, and the other signal would go again to 5 and 0 and 5 again. And then sometimes it will keep at the up time, and sometimes it will skip it. Okay? So basically, if you read the bottom signal, you can say when the line is at 1, when it's 5, we'll say 1, when it's 0, it's 0. And then you have this sequence of zeros and 1, which is a binary code that you'll all know that the computer works with. Okay? So we all know that computers are working in binary manner, but this is not everything. Okay? So on with time, the computers scientists, they developed what they call protocols. Okay, so the first thing, if you measure that, you'll have a really, really long sequence of zeros and ones, but they're not all 0 and 1 have the same meaning. So, for example, the first 4 bits of this sequence will have the version of the protocol that you're working with, and then the next 8 bits is a number, okay? And this number is going to tell you the length of the header. The header is like, so what's going to happen is that you'll first send some information about what you're going to send, and then you're going to send the data. So for example, if you're going to send text, or if you're going to send the image, you might, code the data in a different way. So you're going to say a lot about what's going to happen at the beginning, and we want to know how many bits we need to interpret in order to make the code. Okay, so this is so each one of these bits has a meaning, but if you'll shift the whole thing by one bit, if you'll take one bit and shift the whole thing, all the information that you send to Google, you can forget it, because they wouldn't know what to do with it. It's kind of it's useless, because now the version will have this bit from this part, and then it will be a different version, and then this length will be different, so the whole thing will scramble up. Okay? So you have very, very strict protocols, and in a way this means that if now I will give you, like, I'm now working at the Israeli intelligence, and I'm tapping a computer of a Russian military person in Syria, I'll just get a lot of these, okay? And at the beginning I don't know what to do with it, but since I know it has to follow some protocol, I can start and try to figure out backwards what will fit into this protocol, and I may be able to make sense of what's going on there. It's not arbitrary. This bit has some protocol, and they're following this protocol. Now, why am I telling you all this crap that I don't know much about anyway? Because basically we're doing the same thing in neuroscience, right? We're putting an electrode. So this is a machine called, or I don't know if you can call it a machine. It's a device called NeuroPixel. It's the latest advance in electrophysiology. It's just been released, I think, a few months ago. We're going to get a few labs here are going to get the first one in a few weeks. And this is a very sophisticated device. It's a piece of silicon that on the silicon you have, like, you see every time you have like this black dot, this is an electrode, and the whole thing is built on a silicon. It's just like a silicon chip of a computer, and it has about 900 of those on the chip. So if you put it in the brain, you can record from 900 locations inside the brain the electrical signals, and on top of it there's a small computer that can tell you online, you can choose online which electrode you're going to record from. Okay, so you cannot record from all the 900 at once, but you can select from which ones you want to record and from which ones not. Okay, so this is a very, what we call it, high throughput device, because we can record a lot of signals from the brain simultaneously. And this is what it looks like. It's extracellular, yes. You can make it wireless. It doesn't matter.
But if you connect wires, why won't you be able to record from all of them?
So the problem is always how would you? So imagine I made an electrode like that. I have to connect it to 900 wires, or say 300 wires. This is going to be extremely complicated. So what they did is that at the top of the electrode, they made a small chip, and this chip is doing a few things. First of all, it's digitizing the signals, so then they become digital, and then so first they filter it, amplify them, digitizing, and then they can actually transmit it through one line. You don't need more than one line because it's digital, so it's doing what is called multiplexing.
Multiplexing 900 is just too slow?
Sorry?
And like multiplexing 900?
Yeah, it's too complicated. That's why they're doing 300. But I guess they will just, in 5 years from now, they'll do the full 900. But probably then they will have more contacts. So it's like it's a constant race between what you can do and what's not. Okay, so what you see here, for example, I hope you can see it. You see this like small dip here, right? Okay, so this is probably a spike in a neuron. And since the electrodes are so close to each other, you can actually pick the same spike on several electrodes at the same time. Okay, so you can actually see that this spike would be different than when you see like a dip here, or maybe a dip here, that this looks a little bit different and it's longer, so this could be a different one. So we can classify, so the same electrodes, they can pick up spikes from different neurons, and we can classify which neuron is which, okay, that we're recording from. So this is very interesting, but it's a topic for a method course, not for what we are talking about now. But what I want to say is, imagine that I can translate all that to many zeros and ones, basically the spikes that neurons are emitting in the brain. This is very similar to what I showed you before, right? I mean, it's similar to this tapping to this line, okay? But the problem is that we don't know the protocol. It doesn't matter, you know, if you take the whole 8200 unit and give them the signal. They don't know the protocol. They don't know what to make out of it. Yes.
Not all the physical flow. We don't know the clock, so we can't tell between the
If there is a clock.
If there is a clock, and you don't know, I mean, you ignore the shape, you ignore many things. You just assume that light is all the matter.
Right. So, so what I'm trying to say is that even if we're doing many of the assumptions, we still don't know the protocol. If there is a protocol, there might not be a protocol. So I'm trying to convey. I'll try to convince you now. So this is, for example, what this signal looks like. So what I showed you here is 100 milliseconds, okay, a tenth of a second. But this is what it will look like if you record for a much longer time. And now each dot at each different color is a different neuron after this, what we call spike sorting. And this probe is very long, so you can go from the visual cortex through the hippocampus to the thalamus and record at all these places at once. Okay? And you can give a visual stimulus. You see, this is how it goes. It goes from the visual cortex through the hippocampus and to the thalamus. So you can actually record from many neurons at the same time, and you can give visual stimuli. That's what you see here at the top. Okay, so you can have, like, you can see that there are, you know, repeating things that happen. Okay, but still, it's really, really difficult to make sense out of what's going on. So, yes, question? Okay, so now I'm going to try to convince you that there might be a protocol, or there need to be a protocol. It doesn't have to be as strict, of course, as it has not been designed by computer science. So probably it works in a little bit different idea, but I think the idea of a protocol could be useful. So I want to start with thinking about, we'll talk about it much more later, about these cortical columns, if you want. But just consider this schematic way in which we describe the cortex. Okay, so we know that cortex has layers, I hope. Is there anyone here that doesn't know about layers? I don't mind explaining, and I will explain later, but everybody is okay with layers of cortex? So cortex has layers, and we have this schematic idea that, for example, in visual cortex, and you saw that visual cortex input comes to layer 4, and then information flows in the cortex, but it usually goes out through layer 5, and the neurons in layer 2, 3 are talking to other neurons in other columns in layer 2, 3. So we have this kind of schematic picture in our mind. Now consider one of these neurons of layer 5, and this neuron is sending output to the thalamus. It might send output to the brainstem or to the cerebellum, and it can also send to the colliculus. So it has long-range projection, but we also know that they are talking to themselves. Okay, so if you follow the axon, of one of these pyramidal neurons, so some of the collaterals of these neurons will happen on neurons that are close to it. And some of them will go to a different column. And some of them will go to another column, maybe in the other side of the brain. Okay, to the visual cortex, but on the other hemisphere. and some of them will go to the thalamus. So when you consider now the spikes that these neurons emit, okay, that probably mean completely different things to different targets. Okay, so this neuron, I mean, in our very naive way that we're thinking about the visual cortex, we say this neuron represents a receptive field, and whether the bar in this orientation or that orientation has a preferred orientation, and if we put a bar in this preferred orientation, it fires. Okay? But then these spikes, we have to think, are going to completely different targets. It's going to different type of neurons, to different locations in the brain, with different delays. So the spike will arrive here much before it will arrive there. Okay? And we don't know, and probably here it's going to target a neuron in layer 4, and here it's targeting a neuron in layer 5. So these spikes that we are recording have different meanings in different places and in different contexts. But sorry, I forgot one thing to tell you, which is very important. One of the things that we kind of neglected here is that we have a certain amount of neurons here. I mean, if you look at this scheme that we talked about before, here, then the output of this column is coming through layer 5. Okay, how many neurons do we have in layer 5? Say in a mouse visual cortex. Anyone has like an idea? No, sure number, it doesn't matter. If you're wrong, no one really knows. More or less, 10, 100, 20,000. In a mouse? Okay. It's more like 12,000, something like that. Okay, the whole column usually have something like 30,000. But yes, it's on the order of 10,000. Okay. Now these neurons have to communicate with many different locations. So it's impossible. If you go to this column, you will not get axons from all these 10,000 neurons. Okay? And even if you would, you still have like 30,000 neurons in this column. So there's no way to convey all the information that happens in this column completely, right? You think about this visual cortex column. It has some information now on the visual scene, and this action that goes out, they send this information to Google, right? Like this cable that goes out. Okay? So we need to represent the information in some way and move it about. And because we have so many targets and we don't have so many neurons, we have to do what? What would you do?
Like split the information.
You have this kind of you have like cable of bundles of fibers that are coming out of here, and you need to what you want is to convey the information in the column to everywhere else. So what do you need to do if you have a limited number of fibers? What would you do if you have like a huge text file? You have to send it. You have to send it somewhere, and you don't have a good connection, right? What would you do? You compress it, right? This is what we do all the time. You don't even know that, but everything that you do is being compressed. When you take a picture on a camera, you don't keep all the pixels to your SD card or to the phone, because after 5 pictures, the whole thing will be blocked. You have to compress it. So I don't know if that's true, but it makes sense that a lot of what happens here is actually compression of the information, because we have to send information to so many targets with a limited with a limited amount of fibers. But if we compress the information, then what happens when it comes to the target?
You have to decompress.
You have to decompress. So you have to have some kind of protocols. Okay, you have to kind of represent information in one way when you send it there, and then you have to have as a target something that knows how to decompress this information and make sense out of it. So, I mean, this is completely, I don't think you can read it anywhere, okay? It's a completely personal view of what's going on there. But it makes sense to think that a lot of what happens basically in the brain is just translating in, translating out, compression, decompression. A lot of these things happens all the time. And we need, and if that happens, then the information that is represented in the communication between neurons is probably using different protocols, because the same spike that it goes to the brainstem, or it goes to a different neuron in the same location, it has a different meaning. Okay, and I'll show you some examples in the next lectures on things that we some evidence that we have for these things. Okay. Anyone has an idea what I'm talking about when I'm saying that we have some examples? If I would.
Thalamus?
What about the tunnel?
Compress the information. It's a visual cortex, for example. Retinal ganglia in terms of too much ganglia in terms of the LGN and thalamus. That's compressing all the information.
I'm looking for a concrete example for something that, I have a spike that's coming out of a neuron, and it is translated in a different way. I mean, there are some obvious ways to think about it. Yes. What about synapses?
If you translate from electrical to chemical.
So the synapse is actually translating information, and one of the things that we will I'll show you, I hope you know that, but if you don't, I think it is important to show you, that the same axon that makes a synapse on one cell or on a different cell can have a completely different way to interpret its spike. Okay? We'll talk about it in okay, so let's think about how information can be represented. What are the ways that you know about, that we talk about, and some of it you already talked about in different courses, how to represent information in the brain. How information is being represented.
Rate coding.
Sorry? Yes, what did you say?
Rate.
Rate, what does it mean? It's true, but what we're
Number.
talking about rate coding, what does it mean?
Just the number of spikes in some beam, and it doesn't matter the exact timing of each spike.
Okay, so we're talking about the rates of spikes, how many spikes per second a neuron emit. Yes. What else?
Synchronization.
Okay, so what does that mean?
Just the difference in timing between two neurons.
Okay, so we talk about it, although it's a little bit more vague to say what does it mean, right? I mean, so you have to be a little bit more specific. I'll give you an example about that if you.
We can look at the group of axons of some group of neurons. It's a vector. The combination of spike neuron represents something.
Right. So this is kind of what we call either population coding or identity coding. So the idea is that you take a snapshot of all the neurons and at a certain time, and you say which is spiking, which is not. This is becoming something, a representation of the population of the neuron, and then this will mean one thing, and if you kind of represent it, other neurons will spike and other neurons will be silent, this will mean another thing. Okay. What else? So you didn't. So did you talk about population vectors. We talked about, at least the people who knows what population vector is in the context of the motor system, motor cortex. Well, it's one person, that cannot be true. Everyone that was in the course last year should know what the population vector is. No? So a population vector is a little bit more sophisticated way to represent what you said. So it's not only the identity of which neuron is spiking, but also to what level it spikes. Okay, so it's like a combination of the identity and the rate, and then together we can kind of extract some meaningful information from a population of neurons that we know the response properties of each one of them. So there are also more sophisticated ways. For example, I will talk about it later, which in some ways generalize some of these things that we talked about in a more sophisticated way. So it's not only which neuron spikes when, but what groups, you can think about it as sequences of populations. So one population is spiking now, then a different population is spiking later, and this trajectory in a very high dimensional space, this is what represents information. I will give you some example about it later. So how do we know? I mean, for some of these things we have some examples in the brain. How do we know really what's going on there? So I want to represent now two different ways that we are addressing this problem. One of them is going from first principles, like physicists like to work. Okay, so assuming that that information is represented in spikes, then you can ask yourself, okay, so I have a neuron, what makes this neuron spike? So if I know how spike is being generated, maybe I know something about synchronization, the importance of timing, things like that. Okay, so I can go from the single neuron and try to understand general principles about how spike trains are being generated, and then I can learn something about the code that is being used. Another important issue in this context is what is a noise. I don't know if anyone of you understand what I'm talking about now, but in a second we'll talk about it in much more detail. And once you know these things, for example, what is a noise, how spike is being generated, is a spike analog or digital? For example, does the amplitude of spike have any meaning when you send the information? Then you can start put some boundaries about what code could be used and what not. For example, can you use the specific timing of each spike? And we talked about the clock. What is the rate of the clock? How quickly the clock is ticking? There's another way to look at it, and it's going from the other direction, and this is called like top-down. So, for example, you can give someone some task, and then you can ask yourself, the information has to come from the eye to the LGN, to the visual cortex, all the way through the brain, and then goes to the motor. This is a time limit. It has to be done in that time limit. This puts some limits on the computation. So, for example, if you watch a soccer game, sorry, I'll do it again. Okay, it doesn't matter. So, when you watch this movie, basically in 3 seconds, I'll just do it like now by hand because it's way too fast. So if you watch what happens here, and what I want, usually people when they do that, many people using this kind of example. They want you to focus on the person who is going to score a goal. Okay? But I want you to focus on the goalie, on the person who's actually his task is to prevent this goal from happening. Okay, so if you look at it, okay, this guy is it has to stop the ball here, okay, and then in less than 3 seconds has to come up to figure out with all these like not few number of people who's where the ball is, and what's going to happen, and basically stop it again. So the whole thing is like in 3 seconds, and this basically in a way, and you can, people are doing many things, incredible things. They play the violin, they play piano, they're doing very quick things. What I like about this example is that it doesn't matter how many years you play football, this exact thing that happened here never happened again, before. So it's not like you learned a repeated thing and you have to do it, it's not like you take something from memory and you just take it out. You have to figure out online what's going on here. Okay? So this puts a very strict boundaries about how fast things should happen, and you see that it's not obvious. We don't have infinite time of computation to do the things that we do. We do things pretty quickly. Okay, so I now want to go back to the bottom-up approach, because this part, this top-down, I think it's very interesting, but it's much difficult to follow, and also less relevant to the things that we're going to talk about now. So I told you I want to go into single neurons, and I'm going to do it through the spike statistics. So that's the bottom-up approach, looking about properties of spikes and of neurons, and understanding what we can learn about these protocols from them. So this is a citation already from '69, and it's interesting. I find it very interesting that so long ago people already started to deal with these questions. And it's a speech. It's someone that gave a lecture, a memorial lecture. I put it on the Moodle so you can read the whole thing. But it starts by saying how reliable are neurons. That is, how dependently do they do the same thing under the same conditions. In this meaning of reliability, the question is relevant to or directly raised by much contemporary discussion of the principles of operation of the nervous system as an information machine. And then there's a long list of citations. Early success with stimulus response curve of single neurons, sensory neurons, was so gratifying in the precision of coding by nerve impulses frequency and the tightness of the scatter of points that emphasis was understandably placed on the unit as a discriminating and predictable performance. So the idea was that when people started to work with sensory systems, and when we'll talk about the somatosensory system, I'll give you a concrete example of that. What they were doing, they were taking peripheral nerves, so like axons or several axons that are connected to a muscle, and then stretching the muscle and counting how many spikes are coming out of this muscle. Okay, and then stretching more and counting the number. And this is such a nice system that the idea that came out of it, that neurons are coding the sensory stimuli by the number of spikes. This is where rate code started. Okay, and indeed in peripheral systems, the systems are extremely reliable. So the idea was for many years that neurons are reliable. Okay, and then at the 60s. The pendulum has now swung to the other extreme. The prevailing view, at least among writers and speakers at symposia, regards the brain as basically operating in a probabilistic manner with unreliable components. This view, does it continue? But this is the fundamental boundary that people are kind of, if you ask people, are they right or left, it's the same thing. Some people believe that the brain is completely unreliable, stochastic, and I guess most of what you learn in this neural network courses will come from this assumption. Neurons are probabilistic, they are unreliable, we can't trust them. Okay? But we have examples, and I'll show you some examples, that in some cases are extremely reliable. And this question is extremely important in terms of when you look at spikes, you have to understand whether these spikes are coming from an unreliable source. I have to average them, I have to take the noise out, I can't count on them. Or neurons are very reliable, and when they need to convey some information, they do it very precisely. So let's take a break and we'll continue after.
בתקופה הזאת קראתי ל-ACD הזה, אז לא נצטרך להם יש בעיקר
אני בשיעור, אני רק מסיים עוד שנה.
כל העסק שיחות על מיטות ואיך לישון.
כן, מה המספר עבודה? כן. כן. תודה.
באמת היא נכנסה כאילו לחנייה ושכנה עמדה ממש לידה וכאילו היא לא הייתה תוצאת אותה. אז ביקשה ממני להוציא אותה כאילו, כי אני ממש יודעת לצאת ולהיכנס לחנייה. כן, לא, אני אחזור anyway, אבל מתי אסדר? אני חייבת שתוציא אותי מהחנייה. כאילו מה נתחנייה אולי? אבל עד שחזרתי הביתה הייתי נוהגת, לקחתי מדע שרוני שהיה קורס, ואז כאילו את אומרת מה, וואי, נראה לי שכחתי את נוהגים.
תודה רבה.
לא משהו רציני.
ברוך השם. יואו, עוד אז בא לי כל הזמן. כן, לא,
אני לא במוביל,
וואי,
אנחנו שייכים לאותו גליל, אותם יזמים, אבל משהו אינטימי כזה. לא, אני לא חלק מה...
נגיד הג'ין, מה זה יוגה?
כן, זה גם קשורים.
האמת שיוגה זה מגניב. ייצוג נשים בחרה, היא אומרת לך כזה. נכון, אני גם חשבתי כאילו מביאים אותנו ודוחפים אותנו. זה הלכה להיות. לא, זה לא קורה.
אנשים שעושים בפעולה.
כן, זה גם מה שקורה.
מצלמה? הדור הראשון המחשב יושב בדור של...
ראיתי, נכון,
היית לך מה? אתה בא פעם ראשונה, מה? אתה מדבר שלושה פרצופים, ולפעמים צודק ולפעמים צודק.
בוא נתחיל תפוצה קטנה.
אבל...
Okay. So, so this idea of looking at neurons. So this is an example from a paper already in 1962. It's already before the paper that I showed you. The image is a little bit funny, so I flipped it for you, and so we can look a little bit about data. And what you can see here is what? What is it? Spikes. Probably different electrodes. So you see units, right? So different units, and these are extracellular recording. And why are they so flat here? So they usually high pass filters. So when you measure, you get both the LFP and the spike. So if you do high pass filter, you have a zero average, and then you see these things that are called spikes. So what do we see on these different units? What is your impression? Like, what about this one?
Uniform.
This is quite regular, right? It's ticking almost like a clock. It's not very precise, but like from a biological system, you wouldn't be surprised. Okay. On the other hand, if you look at this one, it's kind of not very regular, right? I mean, you have periods when you have high frequency, then one spike, then maybe silent, and another high frequency, kind of not very organized. So how do you look at this type of data? Some of you probably know, but I want someone that doesn't formally know. What would you do if you want to try to quantify the difference in the regularity between this one and that one? How would you? What would you do?
Change the input. Maybe change the input and see how it change the data.
You're doomed. You put the electrode, you recorded that. You want to say that this one is more viable than that. What would you do to do that?
Maybe the distribution of interest by capital.
Okay.
It's like look at the quantify the density of the spikes during the time.
Okay, so it's basically more or less the same thing in a way. So one thing that we can do is, like, say we take a window, we'll count how many new spikes we have in the window, then we'll move the window, count how many spikes we have in the window, do it again and again and again. And if the window is quite long, what do you expect? And this is like a regular neuron. So what would you expect?
It would be the same.
So the number of spikes within every window will be more or less the same, okay, because it's regular. On the other hand, if we'll do it here, what will happen? It will change, but here it's now becoming a little bit more complicated, right? Because you might expect that if I take a very long window, and this neuron is spiking, I don't know, 10 spikes per second. So if the window is very large, then all this variability will be gone, right? And then if you take bigger windows, the variability will become smaller. Okay? So this is possible. So basically, if we take a small window, then sometimes we won't have any spikes, sometimes we'll have 5, sometimes we'll have 1. We'll have a lot of fluctuations. When we make the window bigger, it will become, the variability will become different, smaller. Okay?
You can check the standard deviation of the interspike interval maybe. And you will see, like, in the second one it will be less than in the first one.
Oh, yes, okay. Exactly. So who's with me and who's completely lost me? Everybody is understand? Okay, so looking at the interspike interval for some reasons that are going back to a different branch of statistics, which is called point processes. So in statistics, there is a branch of statistics that is called point process, stochastic point processes. And stochastic point processes is the idea is that you have a process that at every given time something can happen or not, for example. Okay, so this is like at every given time you can have a spike or not. And they're looking about properties of processes like that. So for example, you would look at a road and a certain intersection, and you look at what is the probability of having an accident in this intersection. So most of the time nothing would happen, and sometimes you have an accident, and then you'll have a period of time with no accident, then another accident, and this gives you like a point process, like the things happen at a certain point in time, and you can look at the statistics of that. For example, it could happen that this correlates very well with rain, or at night. Okay, so this distribution could be very uniform, like every 10 months there is an accident exactly on the day. Probably doesn't work like that with accidents, but it could be something more complicated. So this is ways we can use these mathematical tools to try to understand what's happening here. And one of the useful tools is to look exactly on the interspike interval distribution. So you look at how much time happened between each two spikes, okay? And you look at the histogram of them. So this looks like that. Okay. Okay, let's look at this. Let's look at that for the moment. So this is the interspike interval distribution. So there are very few times where you get 64 milliseconds between spikes. There are very rare events that it takes so long between them. And there are quite a few where you have about, say, 10 millisecond interval between the spikes. Okay? What happens here? Why is it starting like kind of funny? It doesn't start at very low number.
Refractory.
Right. So we have a refractory period. So after a spike, we know that for the next 3 milliseconds, 4 or 5 milliseconds, usually they're not going to happen a spike. So this is shifted a little bit to the right. Okay. So this is a refractory period, and the fact that we have that, it's kind of increased our confidence that this is a single neuron. Because if the distribution would start at 0, this would probably mean that we have 2 neurons, because the neuron cannot spike immediately after it spikes again. Okay? So it's interspike interval distribution, and you can actually fit an exponential function to this histogram. Okay? And what does it mean that you can fit an exponential function to the interspike interval distribution? Anyone know?
Implies that it's Poisson.
Okay, what does it mean that it's a Poisson distribution? Who knows what the Poisson distribution is in a point process? Some of you do, some of you don't. Okay, so a very famous point process like that is a point process which is called Poisson point process, and I'm not going to go into the math of this process, but you can imagine this process as something that you flip at any given time. You flip a coin with a certain probability. Okay, so spike or not spike, and it's kind of completely independent of what happened before. Okay, so just let's ignore the refractory period, assuming that we always have the refractory period, or assuming that our beam size is such that it allows after a spike a refractory period. Then it's just like we never get 2 spikes in one beam, and at every given time we kind of flip a coin again and again and again. So you remember that I told you. as I told you here, that when we take the window become larger and larger, we expect the variability to become smaller. So what happens in the Poisson process is that actually the variance of numbers of spike within a window is proportional to the size of the window, or proportional to the number of spikes that you have in the window. As if the variability doesn't become smaller the longer you get. So if I give you a number, if I give you a window and I ask you how many spikes you have in the window, your uncertainty about how many, you know the average number, but how many exactly you're going to have, you don't know, because the variability is actually very large. Okay, so this is one of the properties of this Poisson neurons and Poisson point processes. And when people started to look at these distributions and saw that they are fitting to a Poisson neuron, this increases the confidence that neurons are very stochastic. Why are they stochastic? Because when we look at a neuron, it doesn't do anything. It fires at a kind of a constant rate. So we have an example of a neuron that fires at a constant rate, and another neuron that trying kind of to do the same thing, but is completely random. At any given time, you don't know if there's going to be a spike or not, beyond the average number of spikes per unit time. Okay, so this is an indication of, in a way, the most probabilistic system that you can think of. You cannot tell anything about what's going to happen except the average number of spikes. So this made people think of neurons as highly unreliable devices. They spike when they want to, completely random. We don't have anything, we don't know much more about them. Some of them are a little bit more complicated. So you see here that you have, in a way, an exponential distribution. So it looks like on short time scale, the neuron is random, okay? But then it also has another peak at some point. So after a spike, within about 150 milliseconds, there is a higher probability of having another spike. So it's kind of, the neuron is spiking in kind of like some oscillatory manner, and within the oscillation it spikes in a highly variable, but it has some long range probability distribution. Okay. So these were recordings from the 60s, and one of the most famous papers about this property was published in the early 90s, 1993 to be exact. And this was done by a pair of two people. One of them is Christoph Koch, who is going to be here. I think in a few months we're going to have a very big conference about, I think the title is What Makes Us Human, something to do with human neuroscience. And Christoph Koch is one of the leaders of the field of consciousness, and he's also the head of the Allen Institute. Who heard about the Allen Institute? What? Who hasn't heard about the Allen Institute? Okay, so you know that Microsoft was founded by two people, Bill Gates. Everybody heard about Bill Gates, but he had a partner, and the name of the partner is Paul Allen. Very, very rich guy. He decided, I think, about 10 years ago to build a neuroscience institute. He paid $1 billion to build this institute. And this is one of the biggest institutes today in the United States to study the brain. One of the things they are doing very heavily is the connectomics of the brain, a lot of mouse genetics. It's basically an industry. It's not so much like a university. It's a factory with many, many technicians, but also a lot of scientists. And the leading scientist is Christoph Koch. So today is kind of big things about the brain. But when he started, he was really focused on single neurons, what they do. And Sofsky was one of his students, and one of the things they've done is they looked at recordings from visual cortex while, I think it was cats at the time, were being presented with visual stimuli. So you might expect that what you saw before was kind of random because I don't know, this neuron is not doing anything interesting. It's just spontaneous firing. Okay? So what they show here is they did this kind of analysis of the interspike interval of the neurons while the neuron has quite sophisticated response to the stimulus. So what you see here is like this is visual stimulus. It's given at time 0, and the neuron elevating its firing rate, just like in Hubel and Wisdom, right? And then you repeat the stimulus again and again and again. Every time you get a different spike train, and then you look at how many spikes you had at time 10 milliseconds, 20, 30, 40, okay? And this is, what is it? This is the firing rate response of the neuron to the stimulus. It's responding a lot when the stimulus starts, and then maybe when the stimulus disappears, and then the firing rate goes to 0. Okay? And in between it kind of has some intermediate firing rate. But you can take these individual spike train and at each one of them look at the interspike interval. Now you wouldn't have a lot of spikes in each one of the spike trains. You'll have like, 10 spikes, so you don't have a lot of intervals. But you can gather them together for many repetitions and then look at what would be the interspike interval distribution at this point where the rate is 2 spikes per second. Okay? Or 0.2 spikes per second. What will be the interspike interval distribution here when the firing rate is high or when the firing rate is low? And for each rate, we look at the interspike interval distribution for this rate. And what they saw is that it doesn't matter what the rate is. You remember that I told you that the variance should scale with the mean when we look at this Poisson distribution. So basically you see that the variance both in V1 and also in MT is scaling well with the mean of the number of spikes. So this neuron seems to be random no matter what the rate of the neuron is. It's not like that when they spike for a long time, spontaneously nothing happens, they become random. Which wouldn't be much of a problem because they don't do anything important. But even when they are responding to a stimulus, they seem to be random with respect to the firing rate that they are spiking. Okay? So all these things led to this idea that neurons, not at the sensory periphery, but in the brain, in the cortex, behave like Poisson processes. And physicists and computational neuroscientists took it quite seriously. And then we can ask, okay, so this is what we observe, but why would it happen? Why would neurons be unreliable? Do we have any reason to suspect that they will be unreliable? What do we know about neurons that will let us think that they are unreliable? They're plastic, but they're plastic on a much longer time scale. So now I'm looking at what the neuron is doing within 10 milliseconds. Nothing plastic happens within 10 milliseconds. But why would I think that I present this to the neuron with the same stimulus again and again, and every time it's doing something different?
The components are variable or noisy. I mean,
What do
the
you mean?
channels, the amount of neurotransmitter release changes.
Okay, that's a good start.
The vesicles that are also released spontaneously.
Right. So we know that there are some factors that, to the best of our knowledge, and this is, we have to take it, again, to the best of our knowledge, some component in the nervous system behaves stochastically. For example, what's that? Right. This is what we call single channels recording. So this is a technique that was invented by Bert Tuchman and Edwin Neher. And you do a patch clamp recording on a small piece of membrane, and all you have in the membrane is a single protein that lets ion in and out of the membrane, and you can record the current going in and out of this protein. And when you do that, and you keep the voltage at different values across the membrane from plus 60 to minus 60, you see that, for example, at minus 40, sorry, minus 50, the channel is most of the time is closed, but as it depolarize, most of the time it is open. Okay, but even when it is open and the voltage is perfectly held at plus 40 millivolts, sometimes in between it closes. Okay, it's not perfectly open all the time when it's plus 40. So what happens there, what we think is happening, is that we have a complex protein, many molecules, and there are random changes in the conformation of the protein, and these are completely random, at least, I mean, to the best control that we can have. We cannot predict when it's going to be closed or not. We can predict on average that most of the time it will be open, but sometimes it closes. Okay? Yes.
I mean, generally speaking, like each neuron is a super complicated system that finds like high temperature, is billions of molecules. I think it's better question why
would it be
shouldn't
reliable?
Why wouldn't it be completely stochastic? And I mean,
so,
like my
so
intuition would
it's
say
a very,
like,
very
yeah,
good
expect
question.
them to be super.
Excellent question. And I don't have a good answer for it, but I can give you like two different views. One is exactly what you said, but on the other hand, you know, I don't know if I'll have time, but I think at least I'll send you the paper to read. I'll show you in a second. It's a bit complicated. Let me see if it's here. I'll give you a short answer and maybe later a longer answer. Okay? The short answer is that yes, the individual components are noisy, very noisy as far as we can say. This is one example. The ion channels. What's the other one? The other one is synaptic release. So we know that when we code with a pair of neurons, presynaptic, postsynaptic neurons, we give a spike. Most of the times we get synaptic release, we get a current going through the synapse. This is time, tempo, voltage flow. Side question. Who thinks voltage flow? Who thinks time? Who doesn't know what I'm talking about? Okay, so we keep the postsynaptic neuron at a certain voltage. Why? Because we know that the current that flows through the channel is depending on the voltage relative to the reversal potential of the synapse. So we fix the voltage of the synapse, the neuron, and we give a spike to the presynaptic neuron. And then we get a current going. This is what we call an inward current. So this is an excitatory synapse that is doing a synapse on the neuron. But sometimes you give a spike. So this is when the spike is given. We give a spike, nothing happens. Why? Because we have probabilistic synaptic release. We have a probability of release. So not every time we have a spike at the synapse releases. What's the average probability of release of a synapse in the brain, in the mammal's brain, mammalian brain, central nervous system, more or less? What? Low. So it's about 0.2, sometimes 0.5. In some very rare cases, it would go to 0.2. And so the probability of synaptic release is low. So most of the spikes that comes to a synapse doesn't actually produce a postsynaptic response.
Is the probability for a single vesicle or is the probability for every vesicle?
So usually so it's again a good question. So usually what we think happens in the brain, in the mammalian cortex, is that you have one vesicle release per synaptic contact. But many times you have more than one synaptic contact between two neurons. So one axon can make different contacts on the same neuron. Okay? So what you would usually get is 0.2 average over all these 4 or 5 contacts. Okay, so the probability for one bouton, for one contact, is even smaller.
With 0.2
0.2
out
is,
of 1.
what do you mean out of
Like
1?
not percent.
Yeah, 0.2, the 20% probability.
20%.
20% probability.
But you said that it can go up to 80%. Like you said, for 0.2 and some 0.8. And
Yes.
so it's like it's huge variability.
Yes, there's a huge variability. Yes, and it depends on many processes that we're not going to get into. But what I want to say is that we have at least 2 major sources of noise that we're completely aware of in the brain. One of them is that when we send a spike, it doesn't always produce a synaptic event. And the other one is that the ion channels are stochastic, which means that if you think of a neuron in the visual cortex, that I present a picture to the cat, and I'm measuring the spike from this neuron, so some of the synapses that are conveying, spikes that's coming from the LGN to the visual cortex, some of them will work, some of them wouldn't. When I present the image again, other synapses will work, other wouldn't. So the input to the neuron is probably different. Okay? Even if all the presynaptic neurons were doing exactly the same thing, the postsynaptic neuron sees a different current. And also the ion channels are stochastic. But what's the other side of this coin?
It should average out the noise, shouldn't it? Like,
We have a lot of synapses and we have a lot of ion channels. Okay, so maybe that's not such a big problem. If we have like, I don't know, 1 million ion channels on a neuron, then who cares that this one decided right now to flip down? I mean, there are so many others that are still flipped up, that this small noise is not going to make any difference. We are talking about big numbers. Each neuron receives 10,000 inputs. Okay? And each neuron has millions or billions of ion channels. So it should average out. So that's the question that's the other side of the question. So we know that there are sources of variability, but maybe they don't play any role. Okay. So what is this idea of the Poisson neuron? So let's forget about the fact that they average out for a second. Let's assume that they don't, and they do produce noise. Then the idea is that we have a neuron, and basically we have a hidden function of the rate of the neuron. So then what the neuron is trying to tell the outside world is some function. This is what I think is happening in the world. This function is what I think is important. Okay, but I cannot send this function because I can only make spikes. So I would flip a coin here with this probability, and if it turns out to be 1, I will spike. Okay, and then I'll go to the next one and I'll flip another coin, and if it happens to be 1, I'll spike. And here I would flip a coin with a lower probability, and then the probability of having a spike here is lower. And this will translate to this spike range, okay, that represents this function. Okay. And then when I repeat it again and again and again, many times, every time I get a different spike range. But they were all produced from the same function. Okay, so now if I average on many of them, many repetitions, or many neurons that are doing exactly the same thing, I can reconstruct back the function. Okay, so this is the idea of how the brain works with stochastic elements, that each one of them is something like a Poisson neuron that has some underlying rate function. Many of the models that the physicists in this institute are talking about are this type of models. Okay? Every spike here is not really important. It's just a sample from an underlying distribution of rate that we cannot see. Okay? Okay. So let's let's make an experiment. Another very famous experiment. This experiment was carried out by two very famous people. One of them is Terry Szymanowski. He wrote a very interesting book about computational neuroscience. And the other one is Zach Mainen, who is a leading scientist of Brain Institute in Lisbon that is called the Center for the Unknown. Okay. Very, very nice place. Very, very influential scientist. But he did this experiment during his PhD, I think, or maybe his postdoc, and the experiment was extremely simple. He looked at pyramidal neurons from the visual cortex, and did a patch recording from the neuron, and he injected a square pulse of current into the neuron. What happens when you inject a current pulse to a neuron? What happens to the voltage of the neuron? It depolarizes. Okay, so the membrane potential goes up. Now we did that with what we call synaptic blockers. So we put all kinds of materials in the bath to block synapses from working. For example, one of them is CNQX. What is CNQX doing? Do you know? CNQX, BNQX. It blocks AMPA receptors. So even if you have AMPA released from presynaptic terminals, it cannot open the receptor, so we don't have postsynaptic excitatory current. We also block inhibitory transmitters, receptors, for example, with materials like gabazine. We call them gabazine. It's one of them. There's another one called bicuculin. It doesn't matter. Okay. It's good that you hear this words and you probably read them as well. Okay. So we depolarize the neuron depolarizes, gets to threshold, emits an action potential. What you see here is 25 repetitions of the same current being injected. Okay? So neuron spikes, then pyramidal neurons have refractory periods, but they don't do this classical thing that they go all the way down to -17, they go up, like in the Hodgkin axillary model. They can, when they return, they can immediately go back and do another spike, and then another spike, another spike, another spike. And then every time you have a spike, you put a threshold, you note when the spike happened, and you see that the neuron, the first spike happened exactly at the same time every time. Okay? But if you go to the last spike, complete mess. The neuron is extremely unreliable here, extremely reliable here.
What is it locked to? The polarization?
This is locked to the current injection.
But it's constant.
So this is constant current, so every time the neuron comes back, it has to do another spike. But it doesn't do the next spike exactly at the same time. It kind of has some jitter. And the next one is kind of increases even the jitter, and when you come to the last one, you can't even tell, you know, it's a complete mess. Okay. So this gives a feeling of maybe a little bit reliable, but mostly unreliable. You know, very stochastic neuron. Okay. Now you take the same neuron, and you take the same current, only now instead of injecting a very constant current, you inject current that is fluctuating. So you take basically a random process, okay, that kind of imitates inputs, synaptic inputs that coming unreliably, in a way like from here, and you kind of sum them up, so you get on average the same thing, but you get fluctuation. And now you take exactly the same current with the fluctuation and inject it 25 times. Okay, and what you see is that suddenly the neuron becomes very reliable. In fact, like it's kind of very rare that at some point it flips, misses a spike and emits a spike at different times. You see it here.
You get the exact same
exactly the same shape of the current, again and again and again.
I mean, why this makes sense? I mean, why the constant current doesn't get the same interval, but the fluctuation current gets the same
It's a
interval?
good question. We don't really know. It's
But
not
I mean,
really
have you
obvious.
tried? No, no, sorry. Do
I
they
have
try?
tried, yes, I have.
Okay. Do they try like a different shape? I mean,
Different sweeps.
yeah, different shape of input or.
Yes, yes. So you can do, for example, what you can do is you can go from here to one of these, but make the fluctuation smaller and then bigger and then bigger and then bigger, and then at some point you get exactly the same thing here. And if the fluctuations are small, like if I put a sine wave here, but with small amplitude, okay, then I will get something in between these two. Okay? So I can go from very unreliable to very reliable by changing the fluctuation.
What happens if you do a ramp?
It's a ramp. It's a very good question. It depends on the neuron. It's actually very strange, but neurons, for some completely other reasons that I'm not going to talk about, they can do quite different things. So you can go, like, for example, from low to high and then from high to low. And some neurons will follow exactly the frequency, and some neurons will actually have what's called hysteresis. So for the same current, on the way up they will fire more spike than on the way down for the same time. Okay? So it depends on the dynamics of the neuron. It's a whole different field that we might touch upon, but not now. Okay. So so now we have kind of an interesting question. First of all, the neuron seems to could be reliable or unreliable, and it seems not so much to depend on the neuron, it's more likely depending on the input. So for example, if we go back to that thing, okay, you can tell me, here, sorry, you might tell me, look, this neuron seems to be stochastic because the input was flat. It wasn't like very fluctuating, or not fluctuating enough to make the neuron spike again and again in the same way. So it seems to me, it seems to be that the neuron is not so much a stochastic thing. It's the input that might be kind of boring or not fluctuating enough. As a side note, I can tell you that you can do a model of a neuron. You can do a very simple model of a Hodgkin-Huxley neuron, but instead of using the Hodgkin-Huxley equations, which are deterministic, so every time you inject the same current, you'll get exactly the same thing, you can change the equation such that they will be stochastic, in the sense that you just assume that you have a lot of channels, like these ones, that behave according to the Hodgkin-Huxley relationship between voltage and open probability. Okay? And we can do a simulation like that. Just put all the channels and then simulate what will happen. And then ask ourselves what will happen. Are we going to get this type of behavior from a Hodgkin-Huxley stochastic version of the neuron? Because then now when we inject a constant current, the channels might flip back and forth. And what's the intuition? The intuition is like what we said before. We have so many of them, it wouldn't make any difference, right? That's what every physicist will tell you. You have billion channels, forget it. But what happened? This is basically a paper that was what I'm telling you now was done by Idan Segev many years ago. And what happened is that right at the point where the spike should start, actually there are not so many open sodium channels. So you have tons of sodium channels, but most of them at this level of -40, where you are close to threshold, most of them are closed. And there are only few of them that are open. And then if you have a little number of channels that are open and they are stochastic, then it's enough to have like few more to start a spike compared to the total number of ion channels. So So it doesn't matter how many ion channels you have, it depends on how many channels you have open and how much can they actually recruit other channels in order to generate the spike. So even though we have we have stochastic elements and we have a lot of them, sometimes this thing doesn't average out. In some conditions, and this is a good example, it doesn't average out, and you can actually generate noise, or you can make a noisy neuron just because it has a stochastic ion channel. And if you want to understand why it doesn't happen here, you have to read the paper. But there is a good explanation for it. Okay. I want to give you another example of, and probably will stop here, although I hope to do much more today, about, just to give you an example, when the system wants or needs to be very precise, it can do that. And I'm not going to go into all the details because I guess Ellie will talk to you about it when we talk about the auditory system. But you know that we have many animals have these property that when they hear a sound at some point they can actually move the head and know in advance or know exactly where the sound came from. And this process is called sound localization, and the best example of animals that are doing it are the owls. Because owls, although they have big eyes, they cannot see so well in the dark, and they rely a lot on their hearing. So this is called the barn owl, and this is a short movie from the BBC, which I do not understand why the volume doesn't. Just play it for me here.
The barn owl locates the direction of a sound in two ways. When the mouse makes a noise, the sound reaches one ear a fraction of a second before it arrives at the other. This minute time difference enables the owl to work out the left-right direction of the mouse. When the mouse is directly in front of the owl, the sound arrives at both ears at exactly the same time. Now it has to calculate if the mouse is above or below.
If you cut just that collection of feathers in the very center here that help the air flow by smoothly, then you get what we've done here to this owl, which is a stuffed owl that's had just those feathers removed, nothing else. So right here you can see that there are two flaps of skin, and those flaps are directly in front of the ear opening. The ear opening is back here. But they're asymmetrically placed. One is higher than the other one. This one is actually lower than that one.
These asymmetric flaps gather the sound from the mouse in different ways. The noise of rustling leaves comes from below the owl. The sound passes under the high flap, which is on the left side of the owl's head, and is reflected by the ruff into the ear. The low flap on the right side deflects the sound coming from below. When the sound is louder in the left than the right ear, the owl knows it's coming from below. By combining the up-down with the left-right information, it pinpoints the mouse with great precision.
Okay. So you probably all know that we are using these time differences between the ears in order to know where the sound comes from. And there are you can also use the amplitude, but I'm not going to talk about it. Really, I don't want to talk about it in details, but there is a model already in 48 that proposed that what you have is an array of neurons, and the sound that comes from one ear arrives on these neurons from this axon, and the information from the right ear comes on a different axon. Okay, and then you have basically different delays. So if the input comes from here, it arrives this neuron much before the information that comes from the other ear, and there will always be a neuron that the information from both ears comes together, and this will tell you, or each one of them will tell you to which direction change the gaze. Okay? And it turns out that if you look at the anatomy, indeed in birds, you have this area called nucleus laminaris, and you can see the axons are coming from one ear, going on one side, and then the axon coming from the other ear, going on the other side, and making synapses on this long stretch of neurons. And these neurons are also kind of looks built for this thing. So they have dendrites, one dendrite going to one side, one dendrite to the other side, and they receive information from one ear from here and from the other ear from here. Okay? You can even have like nice picture here when you see all these neurons. Here, these are the somas of these neurons, and these are the dendrites in red, and these are the axons that come from one side, and the axons that are coming from the other side. So, I'm not going to talk about the details, but what I want to show you is that these neurons are spiking to differences between the timing of the input on order of magnitude of microseconds. Okay, so look, when the input arrives together, this neuron spikes a burst and then 3 more spikes. And if you just change the timing of the input, 120 microseconds. So it's like a tenth of a millisecond. It's shorter than the spike. The spike takes a millisecond. It's like the time difference is so short, and you have a completely different response. And if you change it to the other direction in 90 microseconds, you don't get any response. Okay, so basically the sound differences between the two is extremely precise, and the neurons respond very, very well to these differences. And it turns out so this is an example when the systems need to be extremely reliable, it can make it. But it goes to this, to this length. It's investing a lot of resources because this is extremely important for the owl and for all of us to know where noise comes from. So, and the information is there. So you invest a lot of energy to make a system that will be sensitive to them. That doesn't mean that this is how everything in the brain works. Okay, but it just means that it is possible. So we'll stop here. We'll continue on Tuesday from here on the same subject. And I know that your schedule is still not final, but I hope until the weekend I'll put it on the Moodle with the timing of the exercises and everything. Okay?
So may I ask a question?
So what you see here is basically the number of spikes and the variance of the number of spikes. So if the variance becomes larger, the bigger the average, this means that it's important. That's the average and this is the variance. The sigma square.
So that means that when you measure in V1 and V2.
Yes.
Okay. Thank you.
I don't know who to talk to. It's supposed to be even if it's not now, it's probably from last year, which is kind of at least similar in some way. Let me check who is responsible for that. You know, I will send during the weekend. I will send a list of papers to you. Okay.
שנייה לפני קצת משהו, אבל דיברת על זה שכשיש את האינפוט האחיד, אז מבינים משהו כאילו סטוכסטי יחסית, ואולי זה מסביר למה ראינו את ההתנהגות הסטוכסטית בנוירונים. אז סתם תהייתי, כי אינפוט מהעולם האמיתי שהוא לא כאילו פשוט וולטג' אחיד, אז הוא אף פעם לא יהיה אחיד.
לא, אבל אני חושב שהמסר שניסיתי להעביר זה שהנוירונים האלה, אם הם מקבלים אינפוט שהוא כאילו slowly variable, אוקיי? אז את יכולה לצפות שהם יראו פה, אבל הם יכולים לראות בכל אחד מהמקומות פה בהסתברות פחות או יותר שווה, ואם האינפוט הוא מאוד כזה, אז הם יראו בצורה מאוד מדויקת, אבל עכשיו, יש כאן שתי בעיות. אם את יכולה להגיד, טוב, אבל כשהאינפוט הוא כזה, ואני אסתכל עליו, הוא גם יכול לראות סטוכסטי, כי כאן האינפוט הוא סטוכסטי, ואז אני אסתכל על ספייק הוא סטוכסטי, אבל עכשיו אנחנו בקונטקסט שאני מציג לו, נניח בוויז'ואל אינפוט, אני מציג לו עוד פעם ועוד פעם ועוד פעם את אותו אינפוט. אז אם האינפוט היה עושה את זה, אז הוא היה מאוד רילייבל. אוקיי, זה שהוא לא רילייבל, זה כנראה מעיד שהאינפוט בעצם מתנהג בצורה כזאת, שהוא היה יכול להיות מאוד רילייבל אם הוא היה צריך.
וכאן העניין שפשוט יש הרבה אונסטים,
במובן מסוים, כן, במובן מסוים, בדיוק כל פעם שיש, כאילו, הנוירון רגיש, במובן מסוים אפשר להגיד שהנוירון רגיש לשני דברים, הוא רגיש גם למה המתח, או למה הזרם, וגם לנגזרת שלו. אז אם הנגזרת גבוהה, אז היא כאילו משפיעה יותר מאשר המתח עצמו, ואז...
כאילו שינויים.
כן, אז יש לך פה הרבה שינויים, אז השינויים האלה גורמים לזה שהוא יהיה טיימלופ כל פעם על העלייה הזאת.
ובאמת אם מקרינים גירוי נגיד שהוא כזה ויזואלי, אז רואים מתנהגות פחות...
אני חושב שיש דוגמאות לזה, אני חושב שזה אחד מהדברים האלה שקרו, זה שהם עשו, את צריכה לשאול את אודי על זה, אבל יש להם שם אודי, אני זוכר, זה אחת הפתיחות הראשונות שלי היו על זה, יש להם את הרנדום דוט פרדיים, הדאטה הזה מגיע מהמעבדה של ניוסון, ויש שם את איפשהו אודי עשה את הפוסט. ושם הם היו עושים את הרנדום דוט פרדיים, זה שהנקודות זזות והקוף צריך להזיז את העיניים ימין או שמאלה תלוי לאיפשהו. ופעם אחת הם שכחו להפעיל את הרנדום נאמבר ג'נרייטור, ובעצם הגירוי חזר בדיוק עם אותם רנדום דוט, שלהם זה נראה רנדום, אבל בעצם זה היה בדיוק אותו דבר, והתגובות של הקוף היו מאוד מאוד אליינד לדבר הזה. והם פרשו את זה בזה שהנוירון הוא בעצם הוא לא סטוכסטי, אלא שהוא מאוד זה. אבל אחד ההסברים, אודי למשל טען, שזה בעצם זה פשוט מין אונסטים כאלה, כי זה מין קופץ כזה, אז כל פעם זה מין תגובה שהיא אונסט לגירוי. אז יש עדיין על הניסוי ההוא שנעשה את זה בשנות ה-90, יש עדיין ויכוחים, מה בדיוק זה אומר. No, why you can give something that looks exactly the same, like flat, and the spikes will be
with different interval. Yeah, but if the input is stochastic,
if you have ion channels which are stochastic, the input it also explains why when you have fluctuating input it will be more reliable. In a way, what happens there, one of the things that happens, do you know dynamical systems? So what you can do is you can look at the space between the voltage and the derivative of the voltage. So what happens is that when you have a spike, basically the voltage goes up, okay, and the derivatives go up, so this is when the spike goes up, okay? And then you have some kind of a cycle, like when the spikes go down as the derivative becomes small, and so this is basically the cycle of the spike between the voltage and the derivative of the voltage, okay?
Okay.
Now, when you plot the Hodgkin-Huxley equations, it's as if you have like you have a stable point here. You have a stable point which is the resting membrane potential. You have a stable point here, and then you have a limit cycle which is a spike. Okay. Now, when you make the stochastic, so what happens? What would happen here when you go away from the limit cycle? What you start to get is like a spiral going to this stable point. So what it will look like is like some kind of damping oscillations of the voltage. Okay. Usually you don't see it in Hodgkin-Huxley equations because you go and you make a spike, but you can simulate it and you can find it. So what happens when you have stochastic ion channel is that when the spikes comes back, okay, it can by random fluctuation go here and kind of do that, and then by random fluctuation go here again. So basically you would get a spike, something like that, another spike. Okay.
So inside is the resting state. The stable.
This is a resting state, and this is kind of threshold. It's very close to threshold, but it's a stable point, so it's not going to move from there. So like in Hodgkin-Huxley, you can get cases where you have a spike, and then the spike does here and kind of stays on the threshold, and it doesn't go up and it doesn't go down.
This threshold is not the resting state.
No.
No, this is different.
Yeah. So you have resting membrane potential, and okay. So you depolarize a neuron. Okay. You depolarize a neuron. This is resting membrane potential. Okay. Now you depolarize a neuron, and it does that. Okay. You depolarize more, and it does that. Depolarize more, and it does that. And then at some point you get a spike. Okay. Spike goes down.
Yeah.
Okay. And then if you stop the current, you would go here.
Okay.
Okay. But imagine that you continue the current, so it will go here, and usually what will happen is you'll get another spike.
Okay.
Right. But sometimes, so it depends. You can kind of now play with how much current you get. You would get something that it will do something.
Okay. Okay.
So this is when you go here. Okay. So now when it's stochastic at this region, it can kind of jump back and forth between these two.
Okay.
Stochastically. And then the spikes it can go here, or it can go here, or it can go here. But so you kind of create kind of jitters when the spike happens.
Okay. But if you give a constant input.
This is with constant input, but with stochastic ion channels.
Okay.
Because they can flip you between these two cases.
Okay.
Okay. So this is why with the constant input, you sometimes get jitter when the spike happens.
Okay.
And when you give fluctuating input, it's kind of basically in this region you force the system, so it's much harder for it to go here.
Okay.
Because it has to follow the current that you inject, and the current that the channels are doing is smaller compared to that. So the chances of you don't let it a lot of time to do that.
So if you give a constant input, it will maybe leave the limit cycle. Cycle.
Yes, exactly.
But if it's stochastic, if the input is stochastic, it will maybe it's harder to leave the limit cycle.
Right.